No. Now I'm recording. All right. Now I'm recording. And let me share the screen. I answered earlier a question about the grading. So take a look at your quizzes. I think I've got quiz 0, meaning the extra credit quiz, quiz 1 and quiz 2 graded. All right, so here's the schedule. Today is October 28th. And we are a little behind schedule. We need to finish chapter 5 and then start chapter 6 and finish chapter 6. That's going to be a tall order to do today, but we'll try and get going. There is no lab today, but I will be logging in at 6:30 to 6:45 to answer any questions. At 6:45, I will log out. So make sure you log in by 6:45 if you have questions. You should be working on your unknown project in the lab as well as your infectious disease papers. I think there were 3 students I sent out a test last night. And I believe one of the students has probably already identified their unknown. Okay, so that was a lucky student. All right, any questions about the schedule? If not, let's move on to chapter 5. I think we have a lot of chapter 5 still to do, so we're way behind schedule. All right, so we talked about aerobic respiration. And this slide is showing you a summary of aerobic respiration on the left and fermentation on the right. So now let's turn to anaerobic respiration. In anaerobic respiration, the final electron acceptor in the electron transport chain is a molecule other than oxygen. It can be a nitrite atom or a nitrate atom. This is nitrate. It can be a sulfate atom. It can be iron. There are many different forms of anaerobic respiration, and each form has a different molecule that it sends the electron to. Anaerobic forms of respiration do all have glycolysis, and they all have the Krebs cycle, and I should say the preparatory step too. So they still occur, but they have a different electron transport chain. It's a shorter electron transport chain. So anaerobic respiration does yield less ATP than aerobic respiration. The yield of ATP will depend on the form of anaerobic respiration. Some of them are very good and can generate 36 molecules of ATP. Remember, in eukaryotes, it's 36 to 38 ATP, and in prokaryotes, for aerobic respiration, I should say, they can generate 38. So 36 is really close to 38 ATP. Any questions about that? Other forms of anaerobic respiration are not as good at generating as much ATP, and they will be less than 36, but more than 2. So anaerobic respiration yields between 2 and 36 ATP. Therefore, anaerobes generally grow slower than aerobes because they do not generate as much ATP from a molecule of glucose. Any question about any of that? Let me just show the electron transport chain, try and make that a little clearer. Well, that's not the best one, but that'll work. See if I can blow this up. Yeah, that's better. So this is the chain with aerobic respiration. And some of the proteins are missing, particularly the proteins in the beginning. And that's why they generate less ATP. Any question about that? Oh, let's end this. All right, so let's now turn from respiration, which includes both aerobic respiration, which we talked at in great detail, and anaerobic respiration that we sort of talked only in passing. Okay. Well, now let's turn to fermentation. That is another way that cells can make ATP, starting from carbohydrate metabolism. In fermentation, showing you the overview here, let me blow this up. It always starts off, fudge, got the wrong one. Let's see if I can find it now. There it is. Trying to blow this up so I can see this better. There we go. A fermentation always starts with glycolysis. It then has one or two fermentation steps after glycolysis. In fermentation, the cells can make ATP, but all of the ATP made is in glycolysis. Any question about any of that? So fermentation does release energy from the oxidation of an organic molecule, largely glucose, but it can be another food molecule. And that happens by glycolysis. As you recall, glycolysis previously starts aerobic respiration, and one glucose molecule is made into two pyruvic acid molecules. Two net ATP molecules are generated, and NADH is generated. Okay, any question about that? Let me see if I can get that. There it is. The NADH that is generated is not used by the electron transport chain. Instead, this NADH is used by the fermentation reaction to make NAD+. Okay, why do you think the cell, if it's making ATP by fermentation, reuses this NADH, recycles it in the fermentation, instead of saving it and waiting for it to be used in the electron transport chain of aerobic respiration? For example, your muscle cells can use fermentation and they can use aerobic respiration. Whenever oxygen gets limiting, instead of the muscle stop working, it falls back on fermentation because fermentation does not require oxygen. It does not use the Krebs cycle or the electron transport chain. So why does your muscle cell recycle this NADH, or I guess recycle the NAD plus, and use up this NADH in the fermentation step? Because if it were to keep this and then just wait until oxygen came to your muscle cell, it could turn this NADH to the electron transport chain of aerobic respiration, and generate more ATP than it could in fermentation. Anybody have a guess? Come on, people, somebody should be able to guess. I really appreciate it when people contribute to the class here. You need to make NAD plus, or else glycolysis will stop because of the shortage of NAD plus. And if glycolysis stops, you'll make no ATP. And we already stated there's no oxygen around. That's why your muscle cell is performing fermentation. So there will be no ATP made in the muscle cell. And the muscle cell needs ATP or it can't contract. I'm sure you've all seen in the Olympics, one of the Olympics pushing himself to the limit, and he or she collapses. What happens is they have their muscles are working like mad and they've used up all the oxygen, so they cannot perform aerobic respiration. And then they're falling back on fermentation, so the muscles can continue working because they can't work without ATP. And then they collapse because they run out of glucose in the muscle cell. And then no more ATP can be made, and the muscle cells just stop working. So the athlete literally collapses. And the coach in the wintertime will go out and throw a blanket over them. The heart and the lungs are working like mad, so oxygen and food, meaning glucose from the blood, will be supplied to the athlete, their muscles, and they will then get up and walk across the finish line, usually. Okay, meaning they're perfectly fine. It's just their muscles stopped working because there was no ATP. And that is why your muscles recycle the NADH, because they need NAD plus in order for fermentation to occur. And without it occurring, you can't make any ATP. Any question about any of that? All right, we were talking about the final electron acceptor. In fermentation, the final electron acceptor is an organic molecule. So actually what happens is pyruvic acid actually accepts the electrons flowing from glucose. Any question about any of that? You don't need to know that terribly, but in the making of ATP, the electrons do have to flow somewhere and they go to an organic molecule, okay? And that'll convert the pyruvic acid into the fermentation product, which in this slide is just labeled the end product of fermentation. There are a couple of fermentation reactions, and I'm only going to ask you to know two. There's lactic acid fermentation, which is the one that your muscle cells use. It has glycolysis, making pyruvic acid, and then the pyruvic acid in one step is converted into lactic acid. This is the lactic acid fermentation, which many bacteria use to make fermented foods, such as yogurt and sauerkraut. And the lactic acid helps prevent the food from spoiling. So yogurt, sauerkraut, and what's the other one? Well, sauerkraut, what am I thinking? Cabbage that's been made into sauerkraut doesn't spoil as quickly as other foods. Any question about any of that? The other fermentation reaction you need to know is the pyruvate or pyruvic acid can be in two steps converted into ethanol. Ethanol is a 2-carbon molecule right there. Pyruvic acid is a 3-carbon molecule. So one carbon comes off as CO2, And that is actually why beer has fizz, or yeah, we'll call it fizz. If the wine does not have fizz because it is made or fermented in an open barrel or casket, and the CO2 is vented to the air. If you were to ferment the wine in a bottle and cap it, then we call it champagne. Any question about any of that? A lot of foods use lactic acid fermentation, like most cheeses, cheddar cheese. Oh gosh, I'm trying to remember some other names of cheeses. What is that? Blue cheese. I'm losing all my names of cheeses. Sorry, I'm drawing a blank. Almost every cheese you can think of uses lactic acid fermentation, with the exception of Swiss cheese. Okay, so lactic acid fermentation happens a lot. Yogurt, sourdough bread, most cheeses, sauerkraut. Yeast performs ethanolic fermentation, and that we use to make, well, we do use that to make sourdough bread as well. And sourdough bread, if you don't know, has both bacteria and yeast in it. And we used alcoholic fermentation to make, obviously, wine and beer, as well as sourdough bread. You don't need to know this fermentation here, where propionic acid is made. That is the fermentation, come on mouse, fermentation that is made to make Swiss cheese. And that makes CO2. And if you don't know why Swiss cheese has pockets or air bubbles in it, it's because the fermentation step makes CO2. And that's why Swiss cheese has the holes or the bubbles in it. There are other fermentation reactions. You don't need to know the others, but they're used in industry to make products that we use, like butanol, acetone, isopropyl alcohol, lactic acid, succinic acid, acetic acid. Any question about any of that? This slide, you don't need to memorize this slide, but it's just telling you the different products made in fermentation, the different industrial or commercial uses, and then the starting material, okay? And if you really want to know the microorganism that is used to make that fermentation end product, and the only one I know is Saccharomyces cerevisiae, that we call yeast or bread yeast, is used to make ethanol. All right, any questions about fermentation? I don't hear anything, so I'm going to move on. This table is a good summary of aerobic respiration, anaerobic respiration, and fermentation. Let me blow this up a little. So it has the type of energy producing reaction, the number of ATP that are made per molecule of glucose. And this part of the table you should know. Fermentation generates only 2, and they're made in glycolysis. Anaerobic respiration is more than 2, but fewer than 38. And then aerobic respiration generates 38 ATP per molecule of glucose in prokaryotes and 36 to 38 in eukaryotes. Okay. You probably should know the final electron acceptor also. Aerobic respiration uses oxygen. Anaerobic respiration, I'm not requiring you to remember all the molecules that it could be. In fact, they're not even all listed here. But you should know that it's a molecule other than oxygen. In fermentation, it's an organic molecule. Okay. Any question about any of that? Let me shrink this. All right, I've stated that generally speaking, glucose is the preferred molecule of aerobic respiration. However, any carbohydrate can be catabolized to generate ATP, and the carbohydrate will just be changed into glucose and then sent through the normal way of aerobic respiration. But cells that perform aerobic respiration or your body can digest lipids and protein. There's a problem because these large molecules cannot cross the cell membrane, but we can digest them extracellularly, meaning secrete enzymes outside of the cell to digest lipids and proteins. And that is how we digest lipids and proteins, if you don't know. Microbes do it the same way. They produce lipases and proteases and secrete them out of the cell, break them down into smaller molecules. Like for a protein, you break them down into amino acids, and then bring in the amino acids across the cell membrane. You do need a transport protein. Lipids are broken down into glycerol, and the fatty acids are then cleaved into 2 carbon fragments, which enter the Krebs cycle. Amino acids have a problem. You have to remove the amino group from each amino acid. And then the rest of the remaining part of the amino acid enters the Krebs cycle. So let's show that here. I'm going to try and blow this up. So carbohydrates are just converted into glucose and then sent through normal aerobic respiration. Lipids are broken down into glycerol and the fatty acids. The glycerol joins the glycolysis midway. The fatty acids broken into a 2 carbon chunk and then enters the Krebs cycle. The proteins will be broken down into amino acids, and depending on which amino acid it is, you can join, some of them can join the glycolysis, some of them join, well, most of the others join the Krebs cycle at different stages. The point is we can digest all of our food molecules and send it through aerobic respiration to generate ATP. For those of us who are couch potatoes, and I have to say I'm that now, although when I was younger, I used to exercise a lot, you will break down the lipids or the proteins and then simply convert them into glucose and then send it through aerobic respiration in the normal way. However, for a marathon runner, they can directly digest lipids and send it down this way, but they don't do that until after 45 minutes of their marathon run. Any question about any of that? All right, most cells can perform carbohydrate metabolism to produce ATP. However, there are other ways to generate ATP, and we will discuss that in a little bit. But first, I need to say that we have talked about the different steps as if they were independent. In reality, they are not independent. The anabolic and catabolic pathways are actually joined by common intermediates, and many of these pathways can be amphibolic pathways, meaning that they can function both ways. And when there's an amphibolic pathway, it bridges the pathway between carbohydrate, lipid, and protein, in nucleic acid metabolism. Let me show you down here. Wherever the arrow goes both ways, it's an amphibolic pathway, and the pathway can go in either direction. Most of the pathways are double arrows, and it's not showing you for lipids, but this is a double arrow too. I've already mentioned that we can digest lipids, and we actually send it this way to make glucose, and then we send glucose down aerobic respiration. But as you know, if you eat too much glucose or carbohydrates, we don't store it as glucose or a carbohydrate. We will convert it into a lipid. So we can break down glucose or carbohydrates, and then we make lipids, and then we store it down as fat, a triglyceride specifically. Any question about any of that? The same is true for proteins. I'm not going to discuss it, but as you can see, this one even shows amino acids are both ways going into aerobic respiration. Or we could go from aerobic respiration to make an amino acid. And our cells are not very good at this. Green plants are much better at making all sorts of molecules, starting from a simpler molecule, meaning they're much better at going both ways than our cells. All right, any questions about amphibolic pathway? The point is that it does integrate metabolism, and we have the possibility of degrading a molecule, but we also can synthesize a molecule. And I think, like I said, you all know that. Like when we eat too much sugar, it's more than we can use. We will convert it to lipids and then store it as lipids, a fat. All right. So we've talked about how ATP can be made from catabolism, and we talked at length how glucose can be degraded to make ATP. And we briefly discussed how other molecules can be degraded to make ATP. Well, we can also make ATP from an anabolic pathway instead of a catabolic pathway. This occurs in photosynthesis as well as chemosynthesis, and we're only going to talk about it in photosynthesis. Photosynthesis is the conversion of light energy into chemical energy. The chemical energy is usually converted to, used to convert atmospheric CO2 to glucose. And we call that carbon fixation. This happens obviously in green plants. It also happens in algae, which are green, and then in some bacteria, such as the cyanobacteria. There are a lot of photosynthetic bacteria. Any questions about that? And this is very important because this is how we synthesize organic compounds on our planet. And without photosynthesis, there would not be food for animals and microbes to feed on later. So there has to be something making food, making glucose, and it's mostly through photosynthesis, although a little bit gets made from chemosynthesis and some, what do you call that, ecosystems actually, it might be the major form of making food from chemosynthesis, like a volcanic vent, that ecosystem. That's how food gets made in the volcanic vent. But most of the, on the planet, meaning on the surface of the planet, on the land, most of the energy is made by photosynthesis. All right, any questions about any of that? Photosynthesis only occurs in photosynthetic organisms. They can contain a pigment to trap light and then convert that light into energy. How they do that is they store the energy from a pigment, or not a pigment, a photon of light, and then that's absorbed by an electron, and then the electron starts flowing from the chlorophyll. And then that flowing of the electron is used to make ATP. And I'll show that in a little bit. Usually the light energy is converted directly into ATP and NADPH, but the ATP and the NADPH may be used by the photosynthetic organism to synthesize glucose. And you synthesize glucose from CO2 and water in the presence of light, and the ATP and the NADPH are used in making the glucose. Any question about any of that? What you don't know is, or at least most of you probably don't know, there are two photosynthetic reactions. There's the cyclic photosynthetic reaction and the non-cyclic photosynthetic reaction. In non-cyclic photosynthesis, that's the reaction that makes glucose. The summary reaction, which you need to know, the first one, making oxygen, so it's oxygenic photosynthesis. You take 6 CO2 molecules and 12 water molecules in the presence of light, and you make 1 glucose molecule, C6H12O6, 6 oxygen molecules, and 6 water molecules. Sometimes the 6 water molecules will be erased and they subtract this here. I prefer this equation showing you 12 water molecules, because when we look where all of the oxygens that are generated by photosynthesis come from, meaning there's 12 atoms, 6 times 2 is 12, they come from water. So you need the 12 water molecules. Any question about that? You do not need to engage in photosynthesis to make oxygen, and there are some photosynthetic organisms that perform photosynthesis and they make sulfur instead. You don't need to know this equation. All right, that's non-cyclic photosynthesis, which I think most of you have had some experience with. What you don't know, or most of you don't know, is there's another photosynthetic reaction. And in this photosynthetic reaction, all that happens is ATP gets made. So ADP and phosphate in the presence of light generates ATP. Any question about that? To my knowledge, all photosynthetic cells can perform both photosynthetic reactions, meaning a green plant, when it needs glucose, will engage in non-cyclic photosynthesis. On the other hand, if the green plant cell needs ATP, it'll engage in cyclic photosynthesis. Let's talk a little bit more about cyclic photosynthesis. It starts from light, hitting the photocenter, which is where the chlorophyll is, and 2 electrons come from the chlorophyll. And then these electrons go through an electron transport chain to that protein in the chain, to that protein in the chain, and to that protein in the chain. And then the electrons return to the chlorophyll. Come on, mouse. So the electron returns to where it started from. That is why it is called cyclic photosynthesis. It goes in a cycle. As the electron is moving in this protein here, it gives the protein the energy it needs to pump hydrogen ions from this side of the membrane to that side of the membrane. This hydrogen ion gradient is a twofold gradient. There's more plus charges here than here, there's more hydrogen here than here. These hydrogen ions will want to flow to this side of the membrane. However, they cannot do that except at this channel protein, which is an ATP synthase protein. When the hydrogen ion flows through this protein, it gives the protein the energy it needs to make ATP. And that is how the cell makes ATP in photosynthesis. And this should look familiar to you. It's very similar to the electron transport chain in aerobic respiration. And it's using the same process to make ATP. It's pumping hydrogen ions to one side of the membrane and then using that twofold hydrogen ion gradient to make ATP. You don't really need to know this, but this happens in a chlorophyll. This membrane is the inner chloroplast membrane of a chloroplast. In a prokaryotic cell, this membrane is the cell membrane. So this would be inside the cell, that would be outside the cell. All right, any question about any of this? All right, so we stated that ATP production can have a different energy source. The electron donor can vary with carbohydrate metabolism generating ATP, and that's both respiration or fermentation. Glucose is generally the starting donor, electron donor, but with photosynthesis, it comes from light as the electron donor, and I guess you can say the electrons come from chlorophyll, but light starts the process to generate ATP. The electron carrier can differ, we're not going to talk about that, but the final electron acceptor can also differ. For aerobic respiration, the final electron acceptor is oxygen. For anaerobic respiration, the final electron acceptor is a molecule other than oxygen. For fermentation, it's an organic molecule. And for photosynthesis, for cyclic photosynthesis, I should say, sorry, it is the starting molecule, meaning the chlorophyll. That's both the electron donor and the electron acceptor. Any questions about any of that? All right, if not, that's it for chapter 5. Let's move on to chapter 6. Oops, wrong button. Chapter 6 is on microbial growth. Of course, know the terms, understand the various requirements for cells to grow, understand that there's 4 phases of growth, know what they are, how they differ, what the order is, and then compare the different methods for measuring microbial growth. When we talk about microbial growth, we're referring to an increase in the number of cells, not an increase in the cell size. You can watch a little video of the cells growing, not terribly important. That's different than from children. When we're talking about growth of the children, we're usually talking about the increase in their size, not their increase in number. There are a number of requirements for microbial growth, and this is true for all organisms grow. There are physical requirements and chemical requirements. Different physical requirements include there has to be the correct temperature in order for the cells to increase in number. They have to be at the right pH, the right osmotic pressure, etc. Different chemical requirements. All living things, the cells require a carbon source, a nitrogen source, a sulfur source, a phosphorus source. They require trace elements. They have different oxygen requirements. And some of them have organic growth factors. For example, us, we have organic growth factors are required for us to increase in numbers. That means reproduce. And for children to grow, meaning increase in size. Like vitamin C is an organic growth factor that is required by us. Any question about any of that? All right. Each species has its own temperature requirements. Each species has a minimum temperature it'll grow at, a maximum temperature it'll grow at, and outside of that range, the species will not grow. And all species have an optimal temperature where they'll grow at their fastest rate. You can classify organisms on their preferred temperature that they grow at. So on this, we have the growth rate on the y-axis and then the temperature range on the x-axis. The coolest organisms that grow are called psychrophiles, and they grow at about 10 degrees Celsius. That's the temperature inside your refrigerator. So things can grow in there, and that is why you should throw your food away, unless it's preserved by something else, after it's been in the refrigerator for a week, because the psychrophiles can grow in the food and then cause the food to go bad or spoil. Psychrophiles cannot grow in us because their cells would literally fall apart at our body temperature. There's also a psychotrophs, and they are between psychrophiles and mesophiles. I'll just briefly mention these are the organisms that their optimal temperature is growing at room temperature. So that would be maybe a bacteria you find in the soil, or maybe in the ocean, which most ocean water, well, that's for us. I guess you can have the tropical ocean water, which is close to body temperature. But the mesophiles are organisms that grow best around 37 degrees Celsius, and that's our body temperature. So all organisms that are human pathogens are mesophiles. Any question about that? We also have organisms that grow best around 60 to 65 degrees Celsius. And if you ever see a chicken going around at the grocery store under a heat lamp, that would be about this temperature. And the thermophiles can grow in that chicken. So after it looks like it's been there a long time, it's probably not a good thing to buy it, because that's the temperature the thermophiles like to grow at. And then the hypothermophiles are organisms that grow best around 95 degrees Celsius. These guys were not even known when I was in microbiology. Of course, that tells you how long ago I was in microbiology. But there are organisms that actually prefer growing around 95 degrees Celsius. Remember, 100 degrees Celsius is boiling water. Now, each organism has its own specific optimal temperature and the range which it can grow at. So these categories based on temperature are not ironclad, meaning there is some variety, variance. Like a human pathogen grows best at 37 degrees Celsius, or at least most human pathogens, I should word it that way. But there are some organisms which we consider mesophiles, and they grow best at 30 degrees, like Micrococcus luteus, because it grows on your skin. And our skin is a lower temperature than 37 degrees. And so Micrococcus luteus grows best at skin temperature, around 30 degrees Celsius. Any question about that? Let's briefly talk about food spoilage. Let's see if I can blow this up a little. Within the red zone, which would be about 60 degrees to 130 degrees Fahrenheit, or 15 degrees Celsius to about 52 degrees Celsius, is the danger zone, where bacteria can grow very rapidly. And some of the bacteria may produce a toxin. So if the food has been at this temperature for several hours, and I think it's about 4 hours, the food should no longer be eaten. So if the food is kept, what is that, 15 degrees Celsius, 60 degrees, that's below room temperature, to 130 degrees Fahrenheit, for more than 4 hours, the food should be thrown away. And bacteria can grow slower at slightly elevated temperatures, like 145 degrees, or down to, what is that, 35 to 40 degrees. But the bacteria grow much slower and it keeps better. And as you know, I mentioned in the refrigerator, you do have bacteria growing. What temperature is the refrigerator? It's way down here to 10. But that food can be kept for a week and it'll be fine in the refrigerator. Below freezing, there essentially is no growth because the bacteria are not metabolically active. They may not die. In fact, many bacteria can survive freezing temperature. And then above boiling, very few bacteria grow. And generally above 62, the bacteria that grow probably aren't very toxic to us, and they would not cause us problem if they got in us, because their cells would literally freeze in our body. That's why only the mesophiles are human pathogens. All right, any questions about food spoilage? Food spoilage can also occur if it is put in the refrigerator in too large of a batch, and then it cools too quickly. There is enough time for bacteria to grow enough in the food and then make a toxin. And that can cause food spoilage also. All right. So bacteria also have a pH requirement. Most bacteria grow between 6.5 and 7.5 pH. So in the lab, we'll give you media if you were coming into the lab. If you were to get the media, it would generally be between 6.5 and 7.5 because that's where most bacteria grow. However, there are microbes that do grow outside that range. We'll talk about them in just a second. Usually when bacteria grow in the lab, they will make, in media in the lab, they will make acidic microbial products. And then those acids will inhibit further microbial growth. To minimize the problem of acid production, usually we put buffers in the media so the bacteria can continue growing for as long as there's food in the media. Molds and yeast grow at a slightly more acidic temperature than most bacteria. But there are some bacteria that we call acidophiles, and they grow in very acid environments, such as volcanic environments, where the pH ranges from 0 to pH 3. And this is very acidic. Your stomach is around pH 2, at least when it's producing acid. Microbes also require osmotic pressure. So all microbes require a certain water concentration and a certain salt concentration. High osmotic pressure can remove the necessary water from a cell. And that would be why molasses doesn't spoil. In an isotonic solution, the bacteria lives nicely because water coming in will be equal to the water out. The water inside the cell is equal to the outside the cell. But in a hypertonic solution, there's more water in the cell. And so water will leave the cell and go outside the cell. And that will cause the cell membrane to pull away from the cell wall. And then it'll undergo plasmolysis. And that will inhibit the cell's growth, if not kill the cell. And that's what happens when a bacteria falls into molasses. And that's why molasses can be stored at room temperature literally for years. My mother had a bottle of molasses that we figured was 30 years old, and it still, as far as we could tell, was good. At 20 years old, I actually made my father molasses candy from it, and he ate it, and he thought it was good, and it never killed him, so it still was good. And that was, like I said, 20 years old, had not spoiled. We use high osmotic pressure to preserve a number of food, such as salted meats. I said molasses already. Honey, the same thing. Salted fish. Another one that I only learned about recently, I actually looked up, why does peanut butter not spoil? Because you just keep it at room temperature, and once you open it, bacteria fall into it. It's because of the high osmotic pressure. There's very little water in peanut butter. And when bacteria fall into the peanut butter, the water leaves the cell, and then the bacteria either dies or cannot grow. And the peanut butter does not spoil. Any question about any of that? Oh, some other examples would be candied fruit or salted vegetables, meaning dried fruit and dried vegetables. High osmotic pressure prevents the microbes from growing. There are some organisms which like high salt concentrations, and these we call halophile. Literally, that means salt lover. There are obligate halophiles that require a high salt concentration to grow. And there are facultative halophiles. They do not require a high salt concentration to grow. In fact, they prefer about 2% sodium chloride to grow, but they can tolerate high salt concentrations, like Staphylococcus aureus can grow perfectly fine in 1 or 2% sodium chloride, but it can grow in up to 15% or even 20% sodium chloride. So that would be a facultative halophile. Obligate halophiles will not grow unless they are in a high salt concentration. And an example of a halophile would be the bacteria growing in the Dead Sea between Jordan and Palestine. Those are all obligate halophiles because I believe the Dead Sea has about 30% sodium chloride in the water. Any question about any of that? In a hypotonic solution, that's low osmotic pressure, more water outside the cell than inside the cell. So water will enter the cell by osmosis. And if the cell does not have a cell wall, the cell will lyse, like our red blood cells. If you put them in water, those cells will continue to take in water, and they will eventually lyse. I used to know how long that was. It's about less than a minute. But if the bacteria has a cell wall and the cell wall is intact, the cell wall will only allow the cell membrane to expand so far. And then no more water can come into the cell because there's no space for it. The cell membrane will not expand any further. Any question about any of that? All organisms have a carbon requirement. Carbon typically is half of the weight of a dry cell. So once you remove the water from the cell, carbon is about 50% of the weight of almost all dry cells. Carbon is used as a structural molecule, as an energy source for the cell. Heterotroph use organic carbon sources. Like we are heterotroph, we have to get our carbon from something we eat. An autotroph, on the other hand, can get their carbon from CO2, either in the air or dissolved in the water. A green plant is, for example, an autotroph. Cells also have different chemical requirements. All cells have to have a nitrogen source, a sulfur source, and a phosphorus source. Nitrogen is actually 14% of the dry weight of a cell. Nitrogen is in amino acids, proteins, DNA, RNA, and ATP. So you can see why all cells have to have a nitrogen source. Otherwise, you wouldn't have any of those molecules. Nitrogen is obtained by decomposition of proteins or amino acids by most cells, but some bacteria can actually use ammonium or nitrate as a nitrogen source. And then some bacteria, only a few, can use nitrogen from the air and pull it out of the air. And then we call that nitrogen fixation, where the nitrogen is put into one of the biological molecules. Any question about nitrogen? All right, sulfur is also a requirement. We need it in some amino acids like cysteine and methionine. It's required in certain B vitamins like thiamine and biotin. Sulfur is also used for DNA and RNA synthesis. Most organisms obtain sulfur from the decomposition of proteins, but many bacteria can use sulfate or some hydrogen sulfide as their sulfur sources. And if you don't know, green plants can use sulfate. We have to get it from an organic molecule. Phosphorus is required in DNA and RNA and ATP, as well as all membranes. Remember, phosphorus is in a phospholipid, and that's the main molecule in a membrane. So phosphate, shown here, is an important source of phosphorus. And we can actually digest phosphate. You'll sometimes see it listed on food items. Phosphate can be found in milk and any molecule actually that has DNA or RNA in it. So when you eat something that was once alive, you get DNA, and that will provide you with phosphate. As well as, like I said, phosphate is added to many foods, and you can find it in other foods like milk and dairy products. Trace elements are things that we require in very low quantities. They are inorganic elements like copper, magnesium, iron. And usually we get enough of these, and this is true of microbes also, from their environment, from either the food we eat or the liquids we consume. And the same with microbes. We pick up those trace elements from those sources. Trace elements can be an enzyme cofactor. And as I stated, they're elements like iron, copper, zinc, and we require these. But usually there's enough in our environment for us to pick it up. Selenium is a trace element, which actually in Western Washington and Western Oregon is not enough in our environment, so we need to get selenium from another source. Fortunately, probably most of the food that you get in the stores now is not grown in Western Oregon or Western Washington. So you get the selenium from those sources. And the reason why there isn't much selenium in Western Oregon or Western Washington is because all the rain has washed the selenium out of the soil and sent it down to the ocean, or the rivers down to the ocean. Any question about any of that? So that's a trace element we need to get. And in Western Oregon and Western Washington, it can be a concern if you eat foods which are low on selenium. The same as with iron, well, not iron, iodine, sorry, iodine. We have very low iodine in our food grown in this part of the state. But fortunately, we get food from other parts of the country or other countries, in which case they will be rich in selenium or in iron. And if you're wondering a good source of selenium, just eat nuts. All right, organic growth factors are biological molecules, meaning organic compounds that are needed by an organism or a cell because the cell cannot synthesize that molecule themselves. Examples of an organic growth factor include vitamins, certain amino acids, we call those essential amino acids, and certain fatty acids, we call those essential fatty acids. For example, I mentioned as a vitamin, humans do not make vitamin C, so that's an organic growth factor for us. Humans also don't make the amino acid lysine, so we have to get that in our diet. It's an essential amino acid. And then the purines and the pyrimidines of nucleotides or DNA and RNA, we have to get those in our diet. Usually, we get the purines and the pyrimidines in our diet just by eating something that has DNA or RNA, and then we digest that to get the nucleotides, the purines and the pyrimidines. Any question about any of that? All right. Oxygen is another thing that some cells require and other cells require its absence. So there's a different oxygen requirement that all cells have. I'll word it that way. Molecular oxygen is, in a sense, a toxic substance. And whenever oxygen is around, these toxic forms of oxygen can be made, and they would be toxic to our cells and any other cell, unless they have a means to make the oxygen molecule or byproduct non-toxic. There are 4 toxic forms of oxygen. There's singlet oxygen, where the electron of oxygen is boosted to a high energy state. This oxygen is extremely reactive, and it's actually used, singlet oxygen, by our phagocytic white blood cells to kill foreign invaders. Our body has different molecules which can react with the singlet oxygen and neutralize it, because this would be toxic to our cells as well as to any bacteria cell. Another toxic form of oxygen is superoxide free radical. That is where oxygen has an extra electron, and this extra electron doesn't like staying as one. Electrons like to be as a pair. So oxygen with a negative charge is a superoxide free radical. We get this made every once in a while in aerobic respiration. And whenever this molecule bumps into something, it tends to steal electrons from what it bumps into. and that can damage other molecules or damage DNA, leading to a mutation. Superoxide free radicals are neutralized by cells by the enzyme superoxide dismutase, or SOD. So you have two superoxide free radicals. They get together with hydrogen in the place of superoxide dismutase to make hydrogen peroxide and water. Any question about any of that? All right, well, what about this hydrogen peroxide? Isn't that toxic to cells? Isn't that going to create a problem for the cell? Yes, it does, but the cell has a way to then handle the hydrogen peroxide. And that is the third toxic form of the four toxic forms of toxic oxygen, and that is peroxide anions. The cell can neutralize peroxide anions, or hydrogen peroxide being one example, by two enzymes. You can have catalase, which converts two hydrogen peroxide molecules into two water molecules and oxygen. And the cell, or the cell, it could be and or, can have the enzyme peroxidase convert one hydrogen peroxide and two hydrogen ions into two water molecules. If the cell is in oxygen, the cell has to have either catalase or peroxidase or both. Otherwise, the cell will die from a peroxide anion, such as hydrogen peroxide. Our skin cells have the enzyme catalase in it. And I think, although I don't know this for a fact, but I think all of our cells have the enzyme catalase in it. Some bacteria, most bacteria that live in oxygen have the enzyme catalase, but some bacteria that live in oxygen have the enzyme peroxidase. For example, Streptococcus pyogenes, which causes strep throat, lacks the enzyme catalase. We learned that in our last lab. That cell has the enzyme peroxidase instead. Any questions about any of that? The fourth toxic form of oxygen is a hydroxyl radical. This is a hydroxyl group that has an extra electron, not an extra negative charge, just an extra electron. And anything that has an extra electron is called a radical. Radicals are generally bad, and they are formed at small or trace amounts in aerobic respiration. They are formed by ionizing radiation, or when hydrogen peroxide reacts with certain metal ions. And radicals are bad because they bump into other molecules and then damage them. So a free radical, I'm sure you heard that term, you don't want to consume foods that have free radicals in them because they will do damage in your cells once they get inside your cell. We have antioxidants to help remove free radicals from our cells, either the free radical, hydroxyl radical, or other free radicals. Curiously, vitamin C and vitamin E are two antioxidants that remove free radicals from our cells. Any question about any of that? We have many other antioxidants that also remove free radicals. So that's why a diet in antioxidants is good for you. It removes the free radicals because our cells always makes free radicals at low numbers, especially from aerobic respiration. All right, microbes that use oxygen are called aerobes. Microorganisms that cannot use oxygen and find oxygen toxic are called anaerobes. Remember that aerobes produce more ATP from nutrients and glucose than the cells which perform anaerobic respiration. They don't use oxygen. And so aerobes grow faster than anaerobes in general. All microbes are classified on the basis of the effect oxygen has on their growth. So this slide is showing you the effect oxygen has on microbial growth. If the microbe can only grow in the presence of oxygen, and as you go down the tube, there's less oxygen and there's less growth, then we classify that organism as an obligate aerobe. These cells are growing in a thioglycolate tube, and thioglycolate is a media where the thioglycolate removes the oxygen from the tube. So at the bottom of the tube, there is essentially no oxygen. At the stop of the tube, at the start of the tube, at the top of the tube, oxygen comes in from the environment, from the air. Come on, mouse. And so at the top of the tube, there's full oxygen, 20 to 21% oxygen. And as you go down the tube, there will be less and less growth because there's less and less oxygen in the tube. Is that clear to everybody? All right. The obligate anaerobes are microorganisms that die in the presence of oxygen. So they only grow in the bottom of the tube because that's where there's no oxygen. And as you go up in the tube, it's barely shown here. We should have one dot there. As you go up in the tube, there's less and less growth because there's more and more oxygen as you go up the tube. So that's an obligate anaerobe. A facultative anaerobe is an organism that can perform aerobic respiration, and it will when it can because it makes more ATP from aerobic respiration. So it will have most of its growth at the top of the tube. But as you go down the tube, you'll get less and less growth because there's less and less oxygen. And where there's no oxygen, you will get a little bit of growth because it's a facultative anaerobe. It can grow without oxygen. It either switches to anaerobic respiration or to fermentation when there's no oxygen. So throughout the tube, you'll get growth, but you'll get the most growth at the top of the tube. And as you go down the tube, you'll get less and less growth. There are also organisms which are aerotolerant anaerobes. They are anaerobes, so they do not use oxygen. However, they have all the enzymes they need to detoxify the toxic oxygen molecules so they can grow in the presence of oxygen and they can grow in the absence of oxygen. And they grow equally well throughout the tube. These we call aerotolerant anaerobes. Any question about that? A microaerophile is an aerobe, so it does require oxygen to grow. However, a microaerophile does not grow best at full oxygen. So in the top of the tube, it grows very slowly, if at all. At the bottom of the tube, it cannot grow because there's no oxygen. It grows the best in someplace in the middle of the tube where there's less oxygen than full oxygen. Usually the microaerophiles grow best between 2% oxygen and 10% oxygen. Helicobacter pylori a microorganism that grows in your stomach and it causes ulcers is a microaerophile. It grows best at reduced oxygen concentrations. All right, any question about any of that? If there's no questions, I'm going to stop here, and I will be in the lab from 6:30 to 6:45, and then I'll log off. All right, I'll see you later.